Hello everyone, today we will know about the cytoskeleton structure. So first of all, what is cytoskeleton? Cyto means the cytoplasm and the skeleton means of course the skeleton. That means inside or the cytoplasm a skeleton like structure is present and that is called as the cytoskeleton. So first of all, just imagine an invertebrate animal. So an invertebrate animal. So in the invertebrate animal, that means it doesn't have any vertebral column. So without a vertebral column, so that means the animals should be just like a balloon which is filled with the water. So that means it may or it should shrink like this or it shrink like this because it doesn't have any vertebral column, but it is present like this or they live like this. So why? Because of the presence of the cytoskeleton. So that means in the invertebrate as well as in the vertebrate, the cytoskeleton, it also works like the vertebral column. Vertebral column. How it works like the vertebral column? Because it provides the support to the cell or to the body or to the organisms. So that is the main function of the cytoskeleton, that means to provide the support. And besides providing the support, it also helps in the motility inside the cell, and along with that it also helps in the maintenance in the cell shape. That means the cell shape can be determined or can be possible due to the presence of the cytoskeleton. So now, what are the different types of the cytoskeleton that is present inside our organism? So there are the three types of the cytoskeleton are present. One is called as the microtubule. The second one is called as the microfilament, and the third one is called as the intermediate filament. So these three are the different types of the cytoskeleton that is present inside our body. Of course, these three have different type of the role inside our body. So the main function of the cytoskeleton or the main function of these three cytoskeleton are the support to the cell or to the body. So first look at the microtubule. So what is the function of the microtubule? So the main function is to help in formation of mitotic spindle fiber. Mitotic spindle fiber. So what is the mitotic spindle fiber? So that means, for example, in the cell or during the cell division, so the spindle fiber, let's suppose this is the chromosome in the metaphase state, and in the metaphase state a microtubule spindle formation occur, and this spindle formation and this spindle are made up of the microtubule. So it also helps in the formation of the also helps in the cell division by forming the mitotic spindle fiber formation. The second one is it also helps in the distribution of pigment. So for example, in the plant, suppose the chloroplast, or in the animal, suppose the melanocyte. So they all are the pigment structure that is present in the different organisms. So these pigments are distributed by this microtubule. The third function of the microtubule is the cell differentiation. Cell differentiation. So what is the cell differentiation? For example, let's suppose this is an immature cell. So now it is converted to the, for example, the neuron cell. For example, it is converted to the sperm cell, it may converted into the excretory cell or like the phlegm cell. So that means different cell differentiation are occurred, and it is due to the microtubule. And the fourth function is the cell movement. Cell movement. For example, suppose during the gastrulation, one cell moves to another, and this cell movement is occurred or is provided by the microtubule. And the main function, of course, it also helps in the cell support. That means it supports, it provides the support to the cell. So these are the main function of the microtubule. So now we should know about the microtubule extensively. So the microtubule, it is formed by the tubulin plus GTP or the guanine triphosphate plus Mg+2 or the magnesium. So that means the main component of the microtubule is the tubulin. So please think like the tubulin structure is like that. So that means the tubulin structure, it is a dimer structure. So what is the dimer? That means it has two components, one component, another is the, this is the another component. So this is the dimer. So now this is the one tubulin. So now one tubulin, or to the one tubulin another tubulin is joined, and this joint is accompanied by the GTP with the help of the or with catalyzation of the magnesium. That means with the help of it needs the GTP, and it also needs for the magnesium for the activation of the GTP. So with the help of the GTP and the magnesium, one tubulin joins to another tubulin. So now to this another tubulin are joined, now another tubulin are joined. So like that a strand is formed, and now this strand is called as the protofilament. So now to this protofilament, this is the one protofilament, and to this one protofilament another protofilament is joined. Okay, and to this another protofilament are joined. So like this 13 protofilaments are joined together. So like this is the structure of the microtubule. That means here this is the one protofilament, this is the second protofilament, this is the third protofilament. So like that total 13 protofilaments are joined together, and it forms a tube like structure like this. Okay, so this is called as the microtubule structure. So in the 3D structure it looks like the tube like this. This is the 3D structure of the microtubule. So now if you take the cross section, for example this structure, if we take the cross section it looks like this. So that means 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13 protofilaments. So that means this is the 1, 2, that means 13 protofilaments are present. So in the cross section it looks like this. So in the book you look like this, like structure, and this is the cross section of the protofilament, or this is the cross section of the microtubule like structure. So now come to where the microtubule are found. So the microtubules are found in the cilia, flagella as well as in the centrioles. So first come to the cilia and the flagella. Cilia, another is the flagella. So what is the cilia and what is the flagella? They are the locomotory organ. What is the locomotory organ? That means it helps in the locomotion of the organism. For example, the cilia in the paramecium. In the paramecium, the cilia, this like structure is present and they are called as the cilia. For example, the euglena. In the euglena, a tail like structure is present and that is called as the flagella. So that means the cilia, it is the singular term which is called as the cilium. That means one cilia is called as the cilium, while one flagella is called as the flagellum. Okay. So now the number of the cilia are always high, but the number of the flagella maximum one or the two, or mostly the one flagella is present, or the single flagella is present, or the single flagellum is present, or maximum two flagella are present. For example, in the euglena, the flagella is present. In the paramecium, that means it looks like the slipper. So that's why the paramecium is also called as the slipper-like animalcule. So in the paramecium, the cilium is present, and in the euglena, the flagella is present. So now, in the cilia, or the animals which are the cilia, so that means all the cilia must have moved at a time, so that it can move to the forward or to the backward. But in the flagella, because of the presence of single or maximum to two, that means it is not coordinately mannered. That means it can move independently, while the cilia must have coordinately moved to each other, so that organism can move to forward, either or backward. So now the cilia, it runs like a running manner, or it moves like a running manner. Running manner means all should have, or all runs like this, but in the flagella it presents, or it moves like wave-like manner. So this is the main difference between the cilia and the flagella. So now look at the structure of the cilia and the flagella, or the microtubule which is present in the cilia and the flagella. And but before going to that, for example, this is the paramecium. Okay. So the cilia structure, it looks like this. So that means this is the whole cilia structure. So now some part of the cilia is protruded in the cytoplasm, and some are present in the outside. So the outside part of the cilia is called as the axoneme. This is very important, axoneme. While the part which is present inside the cytoplasm or inside the body, that is called as the basal body. Again, it is the same for the flagella. That means flagella also, it has the same structure. That means the outside flagella structure is called as the axoneme, while the structure which is present inside the body or which is present in the cytoplasm is called as the basal body. So this is the main difference between the cilia and the flagella structure. That means the cilia and the flagella structure, they have two different structure. One is called the axoneme, another is the basal body. And the structure of the axoneme and the basal body is that obviously the microtubules are present inside the axoneme or inside also in the basal body, but the structure are very different. So now look at the structure. So now first we should look about the axoneme structure. So now I am telling about the axoneme structure. That means the structure which is present outside the body is called as the axoneme structure. So now in the axoneme structure, the structure looks like this. So that means here they are called as the peripheral ring. So what are these structure? For example, as we have discussed earlier, this structure. So here, for example, two ring like structure is present. So that means one structure is like that, that means 13 protofilaments are present, and to that again one, for example this is the one tube, and to this another tube are joined, same like structure. So that means if we take the cross section, it looks like this, that means one, another is the another structure. So this is the main structure of like this. So this is called the peripheral ring. So in the axonym, the outside ring are called the peripheral ring, and they have 9 peripheral ring. This is the first, this is the second, this is the third, fourth, fifth, sixth, seventh, eighth, and the ninth. So that means 9 protofilament like structure, the 9 microtubule structure is present in the peripheral, and in the central region, this is the central region, this is the central region, or it is called as the central ring, and in the central region 2 structure is present, one, another is a 2, but here they are present like in the diploid structure or the doublet structure. This is the doublet structure. So the axonym structure, as we, or if we look the structure, the axonym structure, it is designated as 9 plus 2 arrangement. What is the 9 plus 2 arrangement? So 9 means in the peripheral structure, 9 structure or the 9 microfilament structure is present, and which are the doublet. But here the 2, that means the single structure, and that is present in the twice. This is the one single structure, this is the another single structure. That means 2 single, that means it is the 2 singlet. So that means 9 doublet and 2 singlet, and the 9 doublet are present in the periphery region, and the 2 singlet structure are present in the central region, and the all overly it is called as the 9 plus 2 arrangement, and it looks like this. So now this is the central region, and in the central region the central link is joined to the peripheral link by this structure, and this structure is called as the radial spoke. So that means the radial spoke, it joins to the peripheral link to the central link, while the peripheral links, they are again joined by the connectors. These are by connectors. So these connectors are called as the nexin. So these are the connectors, or they connect the peripheral link structure. So now to this, or to this peripheral link structure, a structure like this structure is present. Here the 3 dot, if we see this, so this 3 dot structure, this is main important structure, that is called as the dynein arm. So this dynein arm, it helps in the, or it releases the energy for the movement of the, for the movement of the organism. So that means for movement some energy is required, and this energy is released by this dynein structure. So this is the all about the axonym structure. So now come to another structure, that is called as the basal body. So that means, for example, this is the paramecium, this is the cilia structure, and this inside the body or in the cytoplasm structure, the centrioles are present. They are called as the centrioles. So now in the basal body, the centrioles forms the cilia and the flagella, and the process is called as the ciliogenesis. So what is the ciliogenesis? So that means inside of the body, or the inside, not our body, that means inside the, for example, the paramecium or the euglena. So this structure, so here the centrioles forms the cilia and the flagella, and the process is called as the ciliogenesis. So during the ciliogenesis, the centrioles are called as the basal body. So this is the again another structure of the cilia and the flagella which is present inside the body or inside the cytoplasm. So now the structure of the basal body is different. Look at the structure, and this is same as present in the centrioles. So first, before going to the basal body structure, first we should know something about the centrioles. So what is the centriole? So for example, in the cell or during the cell division, for example in the metaphase stage, they are the chromosome which is present in the metaphasic plate. So now the spindle fibers are formed here. So this structure is called as the centrosome. So now what is the centriole, what is the centrosome? So now if we see the structure, the centrosome, here it looks like this. So this is the centrosome. So in the centrosome, two centrioles are present, and they are present perpendicularly to each other. So they are called as the centriole. So in one centrosome, two centrioles are present, and they are present perpendicularly to each other here. And besides this centriole, some amorphous structure is present, and they are called as the PCM. PCM means pericentriolar matrix. Just imagine, or just like the cytoplasm. So that means inside the centrosome, two centrioles are present, and they are present perpendicularly to each other, and now the PCM or the pericentriolar matrix are present. So now if we look at the centriole structure, let us suppose this is one centriole. So in the centriole, the structure looks like this. Just imagine. So that means in the axoneme, two structure are present like this here. So that means, for example, this structure, that means one tube and it is joined to the another tube. If we take cross section, it looks like this. So here three tubes are joined to each other, 1, 2, 3. So just like that, 9 peripheral structure, 9 peripheral structures are present. So in the axoneme, this is the axoneme structure. So now in the basal body, it looks like this. That means this is the axoneme structure, or this is the basal body, which is similar to the centriole. Okay. So in the basal body of the centriole, the same structure like present, but what is the difference in the axoneme? So the peripheral link are called doublet. So they are the doublet. That means they are present two ring structure, but they are the triplet. So that means they are present on the peripheral ring, same, it is called as the peripheral ring structure, and this is called as the middle part, they are called as the central ring structure, or central ring or central structure, which is also called as the hub. So, in the peripheral ring structure, the triplet microtubules are formed, while in the exonym structure, the doublet microtubule structure are found. And another difference is that inside the central region, there is no microtubule structure, but in the exonym structure, they have two singlet structure are present. So, that is why the exonym structure is called as the 9+2 arrangement, and the basal body or the centriole is called as the 9+0 arrangement. And here the difference is the 9 is the, here the 9 are the doublet, and here the 9 are the triplet. This is the main difference between the exonym structure and the basal body structure. So, now look at the structure here. So, it looks like the bullock cart wheel structure. That means, in the, let us suppose this is the bullock cart, this is the one wheel, and inside it another wheel like structure. So, here the centriole is also looks like same as bullock cart wheel structure. So, that is why the centriole is also called as the bullock cart wheel. This is the very important, and in the previous exam this question was arised that which structure is also called as the bullock cart wheel structure, and the answer will be the centriole. So, this is the all about the structure or the difference between the exonym and the basal body. So, the basal body, it is same as the centriole as in the basal body structure. Here the centriole forms the cilia and the flagella, and the process is called as the ciliogenesis. So, now this is the centrosome, this is the another centrosome. Here in the centrosome, two centrioles are present perpendicularly, here also two centrioles are present perpendicularly. So, now actually first in the cell one centrosome are present, but for example in the cell cycle, this is the M phase, this is the G1 phase, this is the S phase, this is the G2 phase. M phase means division phase, that means mitosis or meiosis. G1 means this is the gap 1. This is the S phase, that means here the DNA is synthesized or the DNA becomes double. So, now this is the G2, that means gap 2, that means the gap between mitosis or the meiosis and to the synthesis phase. So, during the gap 2 phase, so now the centrosome becomes the double. So, the centrosome actually in the cell, the centrosome are present 1, the number of the centrosome are 1, but during the gap 2 phase the centrosome becomes double, just like the binary fission. So, in the gap 2 phase it becomes the double, and now it comes to one end and another is come to another end, and now the mitosis and the meiosis occurs. So, now the spindle fibers are formed. So, this is the all about the centrioles or the centrosomes. So, now come to what is the function of the cytoskeleton. or what is the function of the microtubule? As I have already discussed, but just as a summary I am telling you that one is the spindle fiber formation, the second one is the formation of axoneme and basal body of cilia and flagella. Third one is the it also helps in the cell differentiation. So that means which cell should convert to which cell, and it is determined by the microtubule. Another is the it also distributes to the cell pigment. That means, for example, the chloroplast or the melanocyte. And another is the it also helps in the cell movement. That means during the gastrulation, one cell goes to another, or one cell again come to here. So this is the cell movement, and this cell movement is occurred by the microtubule, and most importantly it provides the support to the cell or to the organisms. So this is all about the structure of the microtubule. So if we like the video, then please like and subscribe my video. Thank you.